You cover it all. You get kind of swirliness. Like it's not real life if you're just teaching.
Swirly, that's it.
It is.
Those are my grades.
Right, yeah. Yeah, that's school, you can really, yeah. That's great price.
Everyone their filters, yeah.
Oh, yeah.
Well, that continues.
You can draw me this and go away.
Yeah, it becomes different drama. Yeah.
Tell me your music. Nice to meet you.
But that's still important. I haven't really figured out.
Well, there's four.
Yeah.
I tried, I was on.
Yeah, so that's something.
Yes, and it's in the blend. I saw his picture. That's what got me. I saw the picture. I went, oh my God. I had him. And the other one, I currently have. What is your? I actually sent them.
Can I trip over one of these tripods?
You could try. I wouldn't, but you can try.
Oh, right. Yeah. That was neat to see that. No representation.
That's
No,
right.
that's right. It had the cutest name. That's so awesome.
And yeah, they always have pictures.
Oh, right.
Right, and I looked at my board. Yeah. That's a return.
That was shit crazy. He's gonna do it. That was neat to see that. Yeah. That's right. Oh, absolutely.
Thank you.
Hello, how are you?
We did good because.
You probably have
Yeah. So that means my cheerleaders and stuff are
All right, let's go ahead and head towards our seats, please. All right. I am Noah Bush, the current KABT president, and welcome everybody. Exciting lineup. I'm not going to take credit for it. Julie Sporting, Drew Ising, they collaborated and put together the program for today, so that's exciting. There it is, yes, high five. And some really cool presentations. Really looking forward to this exciting stuff. I want to take just a minute. Stan brought a group of things. I'll let him say that's all he needs.
As usual, when these meetings come together, I try to bring things that I have scrounged for free giveaway. And the Great Information Center pocket guides are favorites. And there's one brand new on stream fishes. But anyway, I have multiples of these, all 12 of them, plus one that the Fort Riley environmental guys put together, which is one of the finest of all of the pocket guides, equal or even better to the ones that Great Plains put out. The title is Hazardous Plants and Animals. The bottom line is, boys and girls, ladies and gentlemen, stuff is out there. Get used to it, learn about it, but don't panic about it. That's my spin on why it's really, really good. And of course, the key is teaching the students about biology. You don't teach biology, they can teach you. That's 1 minute. That's all I got. Posters from Wildlife and Parks, the three most recent pretty stream things are there and available. The less I have to take back home to Florence. What else is, well, some other, the pollinator thing, which was tough to get. They're little old posters, and I see some of you got them already. Take one. The pollinator thing is a big deal these days. The lepidopterans and hymenopterans. We know that. Go get this.
Hey, I'm under pressure from that there, so it's good. Okay, registration back there at the table. So as if you get a minute or two, you can presentation runner and Mike will take care of you back there. Seriously, for what you get today, the cheapest possible professional development biology option there is anywhere as far as I'm concerned. So if you get a chance when you leave here today, tell people about it and let them know what a great opportunity this was. To be here and collaborate today. I do want to point out the fact that I did bring PA kids during the factory classroom. I'm not really sure what I'm going to do with VIA this year, but I thought it could be an opportunity for us to collaborate via the blog. So if you take PA kids with you today, that's awesome, but the requirement is then you need to comment and respond to a post that I will make. So this year we come up with a lab or a lesson together. Okay, so if you're interested in those, let me know, and I'll kind of give you a real brief rundown. How to get those set up in your classroom. We're planning on that. We'll see how it goes this year. That's it. So I'll let Drew or Julie introduce the speakers, and thanks for being here today.
If you missed anything or you just thought something was really cool, we are currently live streaming the conference, and so we'll have the video up on the website for you to share kind of whenever you need to. There's a link at the top, KABTV at kabt.org you can use. I'm really excited about our first guest. I've done some professional development with him. He is one of the state trainers for the next generation of science standards. He's from Manhattan, Kansas. Jerry Bixby from the Sunset Zoo will be with us first, so why don't you get started on positive concepts.
presentation of the PowerPoint to Julie, and she can give it to everybody as well. So there's a couple of links on here. But like I said, my name is Jared at KABT. I'm the Curator of Education at Sunset City, and I've been working with Matt Crable at the State Department for that. As one of the trainers that goes out and works with a variety of schools on the next generation science standards for the Kansas College and Career Readiness Standards for Science. And so today I'm going to talk a little bit about the using phenomena to make cross-cutting concepts explicit. It's exciting to start looking at the cross-cutting concepts. I think at the very beginning, in the initial kind of launch of the new standards, everybody was really honing in and focusing on the practices, the engineering and science practices. And so now it's nice to see us kind of branching out and moving to the cross-cutting concepts as well, because the cross-cutting concepts, as you are aware, are a really big part of the science standards, as well as the disciplinary core ideas. They all work together, and so leaving one out is not getting at the three-dimensional learning or the intent behind the framework and the standards. So we're going to focus first and talk a little bit about the idea of using phenomenon as the kind of centralizing or the organizing theme for instructional units or lessons first, and then jump into how that can be organized with the cross-cutting concepts, or how that can bring out the cross-cutting concepts, as opposed to instructional lessons and units being driven by the core idea or the topic, focusing in on this idea of phenomenon. So the first thing is, of course, is looking at what is a phenomenon, making sure that we're all on the same page of what that means and what that looks like. So as you guys have transitioned into the standards over the last year or two, have you guys started going this route and really utilizing and really trying to bring out the phenomenon in your lessons? Something that you guys have been working towards and moving on.
Well, this is actually the first I've heard of this, so, and I'm on our curriculum committee, so I think that it's gonna be important.
It's actually, this is something that I've kind of started to transition to, because I've done a couple of different professional development type activities, workshops, things like that. And the concept of phenomenon and using that as kind of the anchoring piece of a lesson really makes getting to the three-dimensional learning much easier. It really, truly does. And so, by definition, the phenomena is something that can be observed and studied, and that is typically unusual or difficult to understand or explain fully. So it's things that happen all around us all the time. It is something that the students can relate to. And we're going to jump into, again, before I get to the cross-cutting concept piece, we're going to get into, what are some examples of phenomena, how to really dive into the phenomena, and how to utilize them. And Brian Reiser, I'm going to throw up some stuff from Brian Reiser. If you're not familiar with Brian Reiser, he's from Northwestern University. He is a leading voice with the next generation science standards. And he's got some great resources out there, and one of them I'm going to be talking about, because I think it fits really good with using and taking phenomena, flushing out the phenomena in your instructional unit, and then how the cross-cutting concepts really fit and connect into that piece. And so, but this concept of phenomena is basically, instead of picking like, oh, we want to focus on energy transfer or something like that, you pick something that is going on, some observable aspect, and that's what you build your lesson around, not around this topic or concept, if you will. And so I'll jump into that a little bit more. This is something that, this is straight out of the EQUIP rubric. Have you guys, is anybody familiar with the EQUIP rubric? The EQUIP rubric was developed by Achieve, which is the organization that kind of led the effort to develop the next generation of science standards, which is now our Kansas College and Career Readiness Standards for Science. So the EQUIP rubric is actually a really in-depth, detailed tool to utilize to evaluate instructional units. So EQUIP stands for Evaluating Quality Instructional Products. And so EQUIP was initially developed for Common Core. It was a multi-state effort that got together with Achieve, and they developed it to evaluate Common Core lessons and instructional products. And now they've transitioned that over and developed one for the for next generation science standards, or our science standards here in Kansas. And so the equip rubric in its front matter to kind of describe the rubric and how to use the rubric, it even pulls out this concept of phenomena and having to use that. The framework mentions phenomena and utilizing phenomena to help with the three-dimensional learning. So this is something that is very important, and it's a tool. It really is a tool to help you get at that three-dimensional learning. So when you're looking at this and starting to explore the phenomena and how to integrate that into your classroom, there are a couple of strategies that people have put out there, and I'm gonna kind of mention a couple of them to help you guys organize and get around to this phenomena idea and start identifying phenomena to investigate in classrooms. So one of the first resources, and again, I'm gonna give this PowerPoint to Julie and she can get it out to you guys so you guys can visit these links. But this is from the teaching channel, a blog post from the teaching channel website. But this is a nice step-by-step kind of way to really look at phenomena and identifying phenomena. And some of the other things, some of the other resources and strategies that we're going to mention next really kind of fit into this whole overall process of identifying the topic, really looking at that. So when you're using phenomena as you're organizing piece with instructional units or lessons, it doesn't mean that you disregard the topic or the concept or the core idea. But it's a way to get at that core idea in a more meaningful way, but also bring in those practices, cross-cutting concepts, things like that. And so the phenomena, looking at it from the phenomenon picture, big picture, it really helps you organize your thoughts and getting at the three-dimensional learning. So looking at the topic, the standards, because we all have the core ideas that you guys are connected to your guys' classes that you teach. And so looking at the topics, and then starting to take from that big picture, starting to walk backwards and looking at, well, how does that connect to the real world, the everyday world, and what's going on around them? Whether that is things that they're observing at school, the kids are observing at school, at home, in the news, wherever it is, how can you connect that topic, that core idea, back to the real world? And so this is kind of what it's looking at with this phenomenon. And there are a couple of things, and like I mentioned, it's looking at drawing on everyday life experiences. And so, if you're looking at, resource conservation, things like that, you can look at drying hands. There's been, I don't know how many conversations I've been in with people in the bathroom about, the paper towels are worse for the environment, or the hand dryers are worse, or this hand dryer is better, or that hand dryer is better. It's like, well, who's investigated that? Who's done that? And so if you're looking at resources and resource conservation, then look at that. I mean, that could be a real world phenomena that you take and you investigate. And so that's an example of taking where real life experiences, or when you're looking at genetics, the inheritance, the traits, things like that, looking at, I've been told that I have my son has my smile. Okay, well, we have the dimple, everything like that. Well, why? Why is that? My daughter doesn't have the dimple, but my son does. How did that happen? And so you start looking at these things that are real to them, and that's that phenomena that you build into, and you have to break those phenomena down into each little area and what they need to know to understand that phenomenon fully from the science perspective. And we'll get into that with Brian Reiser's resource. Again, you can go bigger. The media, looking at the media and how and what the news that they're hearing or the news that's out there and how that relates to science, the science core ideas. And so drawing upon that and making it real world for them. I know we mentioned the pollinators, so the colony collapse disorder, things like that. Looking at the pollinators, that is definitely in the news. And what they're interested in, so food and clothes and allergies, things like that. I mean, those are things that they're interested in or things that are impacting them directly. And so these are all just ways that, and areas that you can go to try to identify some phenomena. Another thing you can do is Project 2061 They have basically listed out a bunch of phenomena on their website. And this has been up for quite a while. It's not something that was posted with the framework or the development of the framework or the standards. And so this has been up there for quite a while. And they've got a lot of great examples for exploring phenomena and what are some phenomena that you can explore with your students. And so those are some examples. One more example, and that's what I was going to get to, is Brian Reiser from Northwestern University. But he's got kind of two examples that he uses that he regularly uses to kind of show the difference as far as the topic and investigating a topic versus investigating a phenomenon. So, for example, after rainfall, how does water on the street dry up on a sunny day? How will water dry up? Versus how does evaporation happen? So investigating what they're seeing and what they're experiencing versus we're going to investigate the science concept or the core idea. And so that's one of the key things, in going the phenomena route allows you to break down what they're learning and reach those three-dimensional, the three-dimensional learning identified by the standards much easier or much better. And so this resource that Brian Reiser has developed, it's called a storyline diagram. He's called it a storyline diagram. But this is what it looks like. And this is basically, you have your anchoring phenomena. From that anchoring phenomena, you develop this driving question, your essential question, and then you break that down, and that helps you get at these three-dimensional learning, because you're starting to look at, okay, what does it really take to understand this phenomenon? Because a lot of times what we get into is the rut we get into, or the habit we get into, is we start looking at this phenomenon and we don't break it down into the individual little components that they need to know to be able to truly understand the phenomenon. And so this example, this is a good example of that. And so, you know, this is kind of a phenomenon. We run out of energy at the end of the day. We feel tired right before lunch. We have more energy after we wake up in the morning. Working hard makes us sweat and breathe harder. So all these components kind of go together into this phenomenon. And so some initial ideas, some thoughts that we come to the phenomena with, the students might come to the phenomena with. And then here's the driving question. What is going on inside our bodies that helps us get energy to do the things we do? And so there's a lot of components in here. So what are some of the core ideas that that a student would need to know to be able to answer that question? Give it off for a second. What are some core ideas that a student needs to know to be able to answer that question? What are some different things that they would need to know or learn about?
Respiration.
Respiration. Digestion.
Homeostasis.
Homeostasis.
How enzymes work.
Chemical reactions.
Muscles on a cellular level.
Okay, so cellular level. The cell structure, this parts of the cell to be able to do what it needs to do to get the energy. Process that. So yeah, so there's a lot there in this one phenomenon that is happening all the time. And it is something that they can relate to, the students can relate to, but it's not just investigating the structure of a cell to investigate the structure of a cell because it's in the standards, we think they need to know that. It is investigating the structures of the cell in a way that gets to how their body works in an everyday situation. They're in sports. They're working a job till 11 o'clock at night, cleaning dishes, and then they go to bed and they wake up in the morning and they're tired and they come to school. So having those understandings of how their body works, that's what these phenomena help you get at. And so using the storyline and breaking this down in the storyline, you've got the anchoring phenomena, you have that driving question there, and then this is just kind of a breakdown of some of Brian Reiser's initial thoughts on, okay, where would I go? How would I go from this anchoring, driving question to investigate this phenomenon, and how would I go to get to what I want them to understand?
May I ask you a question?
Yes.
Or it may be an observation. It seems kind of like this is what we used to do at the end, and now you want it at the beginning. Is that right?
Yeah, I mean, essentially, yeah, because you're wanting them to investigate. You're wanting to use with the idea of the phenomena. You're wanting that to drive what they're learning, and so you don't want to present them a bunch of different stuff and then have them connect it to the phenomenon at the end. You want them to use that phenomenon and then break off and start investigating different components and different core ideas with the understanding that they're investigating that so they can ultimately answer this question.
Okay.
So this is just one thing that, this is then what Brian, after going through this, Brian Reiser kind of put together this example argument statement that then a student might come up with or could potentially come up with after going through this as an instructional unit based on that storyline. And so this is just something I'll let you read through that, but this is an argumentation that they might come up with after going through that storyline and the instruction that it would follow through there. All right, so the idea of the phenomenon and using that, it really allows, so if we go back and look at the storyline diagram, you know, it brings in and allows you to start looking at, okay, well, what are the practices that we're going to need to do to be able to investigate this phenomenon? What are the core ideas? There's multiple core ideas that are connected to this. And then the cross-cutting concepts, where does that fit in? And so that's what we're going to look at kind of next, and how this exploring, developing instructional units around the idea of the phenomenon. And you know, the practices are there, the core ideas are obvious, but where do the cross-cutting concepts fit in? And so what about the cross-cutting concepts? Well, the cross-cutting concepts are kind of this big overarching piece that connect everything, and it's kind of like our organizing our filing cabinet or filing system for understanding science. And so you have the patterns, because patterns are around there, and so you start making inferences and patterns, the cause and effect. So when we're looking at phenomena, it drives you to ask cross-cutting concept related questions, because you have to define what you're really going to be investigating. So you're defining the systems. You're defining the cellular system, you're defining the digestive, the respiration. You're looking at, you have to define all those systems and at the different scales, at what you have to look at to be able to make sense of that question and be able to answer that question. So when you're looking at it from the phenomenal standpoint, you have to ask those cross-cutting concept questions like what is the system I'm studying? What is the scale of the system that we're studying? What is changing in the system? What is causing change in the system? And are there any patterns? Those are some really basic, standard, cross-cutting concept-oriented questions that you ask when you're confronted with a new phenomenon. You ask those questions, or at least from a scientific perspective and a scientific mindset, those are the questions that we come up with and we start looking at when we approach new things. These unusual or novel experiences, those are the questions that we look at. And so by going the phenomenon route in your instructional unit, it forces you and it forces the students, if it's presented to them in this phenomenon standpoint, it forces them to ask these questions, or for you to help them understand that we need to ask these questions first. And then as you start doing that, you use the terminology. You use that terminology. So what is the system? What are we studying here? Okay, well, and the driving question that we're looking at, the system is really the human body is what we're looking at, but there's lots of little components to it. And so what's the scale of each of those different things that we need to look at? And so you're looking at this phenomenon and you're asking these questions to help the students look at that. And then as you continue to use phenomena in your instructional units, they're going to start asking the same questions. And you're going to start asking these same questions to have them investigate and to have them look at these novel experiences from that scientific mindset. And so it's really looking at what are the cross-cutting concepts and how do we define what they're experiencing through the cross-cutting concepts? Because that's the whole idea behind it. So in here, we identified the different systems that we needed to look at within the human body. You guys brought those up when we first initially talked about. We're looking at energy. And so that is something that we need to look at. We're looking at energy and what our current understanding is of energy. And so there's a lot of things that's just in that dialogue as well with the cross-cutting concepts that you can get kind of a baseline of where the students are at as well and their understanding. But it's really that cross-cutting concept that is going to define and push forward and drive forward the instruction and where they're going next. And so that is where really the phenomenon and incorporating the phenomenon, that is where it really kind of drives with the three-dimensional learning, is that. And so, as we move forward with implementation of the standards, that is a key component and it helps, it's a tool to help make things easier. So what I want you guys to do is, you guys are nicely organized at the tables. So I'm going to put this back up here, and the anchoring question, so in your tables, what I want you guys to do is to go back and write down the anchoring question, what is going on inside our bodies that helps us get energy to do the things we do. And so at your tables, how would that, if you're looking at that driving question, how would that look in your classroom based on this storyline? Because there's no right or wrong way to develop these storylines like Brian Reiser had here. There's no right or wrong way to do this, but you need to ask those basic cross-cutting concept questions and figure out what are the steps. How would you go about doing this? What would it look like in your classroom if you're looking at that driving question? So I want you to take just a couple of minutes and at your table, ask those cross-cutting concept of that or questions of that driving question and come up with your storyline, really focusing in on the green boxes there for this activity. But what is your storyline for the driving question? So go ahead and take a couple minutes and work together at your tables and see what kind of storyline you can come up with.
What was the rest of the question?
Well, I think that the idea. So what I'm thinking is supposed to be passed around.
No, it's just for you. You said you wanted it.
Yeah, I'm trying to figure out. I'm on there. Is there a hashtag?
I've been doing hashtag KABT 2015. 2015.
Yeah, I thought we're looking at like the questions and phenomenon.
We're using that driving question.
The driving questions.
What's going on in our body?
This reminds me a lot of
So how do you see that? So scenarios, phenomenon.
Yeah. So
But what could you actually do?
yeah. So that our model is more complex, so has how to do the questions, how to solve it.
Yeah.
That's hard.
So if you started out with that, 'cause I don't know.
I feel like you might get those from your students. Well.
But if you don't start with it like that.
Yeah, I just want to play with it. About 1 minute, we'll run for discussions.
So, I guess we're coming up with questions to get those initial ideas. So, I guess if it's, we're not picking the ones that's there,
This is where we start to get more into like what we're actually going to teach, like with like a cellular aspect and what enzymes do, what happens in
yeah.
the mitochondria. Or, and then we start going on what we're teaching.
Well, I think that the way it would work is that you have them, like, if this is what I'm thinking it is, where it's kind of like argument-driven instruction, which is kind of becoming more and more into what we're doing. It could be something where we're using that driving question, we give them some information, some data to work with, some research, or they have to set up a lab to do something that involves this. Or maybe they test. Well, can you figure out a reason why you start breathing faster? Why you might need that? Have them get into that and form an argument based on it. And then you get all these initial ideas. You take those initial ideas, and that's the point where you can teach from there. And maybe look at those things one by one, and then have them revise arguments and whatnot.
Alright, so let's, in your guys' discussion, did anybody, was it a lot different than this storyline? Some of the things that you decided to talk about and focus on? Is there anything that you added in that's not kind of listed up here with the storyline?
It's not really added in, but there's a pretty good discussion about how teachers are starting to use a lot of argument-driven instruction because it does such a great job of doing all of these things with the students and giving them their own kind of individuality still as they're trying to connect the data to the conclusion they're trying to draw from it. And you still get the chance to hit all these really important practices.
Yeah, definitely. And that's what I mean, when you're starting with that driving question or whatnot, you really are trying to develop the evidence to lead the argumentation aspect of it. And so having that, I mean, the questions are really the key to it.
We actually went back a step before that to have them come up with the phenomenon by giving them the assignment that they were to record all their food and drink and energy levels over the course of like 24 hours and then come back in and brainstorm what was happening between them and even try and get them to come up with the original questions in the phenomenon in the first place.
Yeah, definitely.
I think too, along that same note, we talked about, I've traditionally used this kind of this exercise lab, basically gather data, show homeostasis and respiration, heart rate and all those things. But if you did that at the beginning and had the kids, exercise, and then as you go through the lesson, then, let's look at this data, how this data. So basically you're creating this model, creates the background of the prior knowledge. So the kid already has something to draw on. They have this, okay, so when we did this was happening for our bodies.
Yeah, there's definitely multiple entry points into doing this. There's not, like I said, there's not, one way to do this or do the investigation. It's really getting at looking at their experiences or things that are going on around them, that phenomenon. And then how do they define that phenomenon? How do they start investigating that phenomenon. And that's where those cross-cutting concept questions come out. And that's how, using that terminology of, defining the system, what are we looking at? And making that terminology very explicit as you're going through that. So then that as they continue to investigate phenomenon, then they're going to continually see that as this organizing concept that they can wrap their mind around and start making inferences and whatnot using the cross-cutting concepts, because that's going to really bring those out. And so that's going to, there's no real perfect way to jump into it or start it or look at the different phenomenon and how you introduce it. It's going that phenomenon route as opposed to let's look at the cell structure or let's look at the digestive system or something like that. Well, why? Why are we looking at it? What is going on around them that makes them know that they should be looking at that, or they should have an understanding of that? And so that's that phenomenon, and then that phenomenon brings in that three-dimensional energy much easier. I think I'm getting close to time. So, like I said, there were a number of links on here. So I'll get that to the PowerPoint to Julie, and she will send that out. The PowerPoint also has my contact information, so feel free to reach out, ask questions, anything like that. If I don't have answers, I can forward that on to any of the other KSD trainers as well, and they can help get answers to your questions, because we're all here in this together, implementing it, whether it's high school, middle school, elementary, informal. We're all launching this and jumping into this head first together. So we all just need to work together and work through the challenges together and ask questions as we go. So anyways, thank you for having me out here. I enjoyed it, and I hope you guys got something out of it, and enjoy the rest of your conference.
Thanks again. Like he said, I don't know how much longer it exists, but there is some funding at KSDE that pays for people who are certified as these trainers to actually get a substitute teacher or to take the day off of work to come to your districts and do presentations for you. So for a relatively nominal fee, the district, I don't even know if there is one at this point, you can get some pretty good professional development for a half day or a whole day, and not just a half hour. And we've got things we can show you, we can actually walk you through activities that do this, and we got some pretty good training. So it was good.
I think Matt did say that they did get some budget approved again for this fiscal year for that. So I don't know how. But
so spend it.
Yeah.
Quick.
So reach out to Matt, and he can coordinate that and get you connected and how to take advantage of that. But yeah, there's trainers all over the state, and so it doesn't matter where they're at, but they can do it.
Our next presenter is a faculty member at the University of Kansas, Ms. Peyton Schultz. She's with environmental science.
And
Okay, in the campus. She went to real life. She's going to talk to us about soil communities and plant diversities and prairie reconstructions, restorations, and how to take those experiments and then kind of take it into educational outreach at the same time. Okay, so I am actually really excited to hear this presentation. I'll get out of the way.
So, I am new to Kansas. I just moved a month ago from Indiana, and where I was working at Indiana University for about 15 years. And the focus of my research for more than 15 years was prairie restoration. So let me just try to get out of this. And so what I'm going to do is introduce you first to an ecosystem I'm sure you all know a whole lot about, and then talk about some of the research that I've been doing. Not going to give you a whole lot of graphs and data slides. If you want to see those, if you're interested in those, I'm happy to show you later. And then talk about the research, the outreach that I have been doing with students. Not so much research about education, but just trying to work with students at many different levels to get them engaged in science and questions about science, asking questions rather than sort of just learning the vocabulary of science. So my job was at Indiana University was to teach environmental biology, which I did to non-majors, juniors and seniors at Indiana University. And then I was also the director of outreach. So I developed programs to take into elementary schools. I also developed programs to work with high school teachers and faculty to develop experiments teachers would take back to their classrooms. So that's what I'll be ending with. Okay, so isn't this pretty? This is a rendered prairie right along the interstate, right outside of Chicago in the north. So in the easternmost part of the Prairie Peninsula, the prairie is very beautiful, diversity is very clear, it almost looks like a garden. And I'm sure we can see that here in Kansas too. So this is Illinois, which is where I began working on prairies. And this is Indiana, northwestern Indiana. And this is one of the most pristine prairies that we see in Indiana. And it has something, a phenomenon here that perhaps some of you recognize or gives you a clue to where we find prairies in this easternmost part of the Prairie Peninsula. Anybody recognize something in the background there?
Cemetery.
So cemeteries and railroad sides are the places where we find remnants, and generally they're very, very small. So the easternmost part of the North American, the centralmost part of the North American continent, up until about 150 years ago, was mostly this, and now only small hectares, especially in and around Chicago and northwestern Indiana, are prairie. Most of it's gone, now it's agriculture. So for the last 10,000 years, the establishment of prairie and maintenance of prairie happened in conjunction with the retreat of the last glacial maxima. Over time, these grasslands developed and they were maintained. They were the plants they were selected for and maintained by two major forces. One is grazing. Lots of different organisms graze on these plants, and fire, which were caused by lightning strikes and were also set by Native Americans. So the plants in this ecosystem were able to survive, were selected for and able to survive because they have massive root systems. These root systems can extend meters down into the soil. And many people talk about them as upside down forests. Maybe those folks out in the East Coast. But so 2/3 of the biomass of these plants are below ground. And so when a grazing event happens or a fire event happens, the plants can take advantage of the energy that's stored in their roots, and they have a growing tip just beneath the surface of the soil. So after a grazing event, after fire, they can resprout and grow. So while these plants can survive grazing and fire, they were much less able to survive plowing. And so in the mid 1800s, John Deere invented the steel blade plow, and that was what caused the loss of this ecosystem. 98% of the prairie and easternmost part of the Prairie Peninsula was plowed underground. So when you look at this land that was prairie, and then it was plowed, and then what happens after we abandon it? It's abandoned for whatever reason. What we see is an encroachment of forest, and we see a lot of introduced plant species. So very few of the natives remain. There are some natives, but very few of the prairie natives remain, in part because the seedbed isn't there. When we restore these sites, what happens? Well, what you're looking at here is a picture of one restoration at the Fermilab National Accelerator in Batavia, Illinois, where we began some of this work. And so, does this look like what you saw before when I showed you this? It is a prairie species. This is big blue stem, but it's mostly big blue stem. So the dominant species we can restore. We're getting those dominant species back with just sowing seed. But the diversity that we see in remnants is not there. And so we began this work more than 20 years ago asking the question, so when we have this transition from prairie to agriculture, and then movement to post-ag, and finally restoration, why is it that we don't see the same diversity that we saw originally in those prairies? And so we asked the question, do components of the soil microbial community mimic the establishment, the re-establishment of prairie plant species? And I'm going to introduce you to one of the organisms, or one of the a group of organisms that we have been working with for many, many years. And these are arbuscular mycorrhizal fungi. So you wouldn't see these above ground. These fungi associate with the roots of host plants. And so what you're seeing here are clear roots. So we've gotten rid of all the material. What's blue is the fungus for the most part. So these are the arbuscules. So this is a mutualistic relationship. The plants provide carbon to the fungus, and the fungus pulls in nutrients and water, perhaps. It's certainly in certain situations to the host plant. So it's a mutualism. These arbuscules are the sites of nutrient and carbon exchange. And these vesicles, if you can see these sort of oblong circles, spheres, those are the resting structures where the fungi sort of hold nutrients or hold fat, really, is what it is. So storage structures for energy. And then these are the spores. So they don't form nice big mushrooms and fruiting bodies above ground. They mostly form these naked spores in the soil that you have to see through extraction of soil. It's not hard, you just use a sucrose gradient, but these spores are tiny. They vary in size from about 15 microns to a millimeter in diameter. So they're pretty small, but they are very big. And please stop me if you have any questions.
There's an arbuscule.
An arbuscule, that is a tree-like structure that is in, so this is a cell.
Right.
Okay, so an arbuscule is a site,
A sporangium,
it's just part, it's a,
a spore forming body.
so the arbuscule is a structure that forms within the cortical cells of the plant for these AM fungi. And it's the site of nutrient and carbon exchange. So it's where the plant gives carbon to the fungus, and the fungus gives nutrients, mostly phosphorus and nitrogen, to the host plant.
But it's not cellular.
It's within the cell.
It is
cellular.
cellular. Is it?
It's
it
a cell by
organelle?
itself, in and of itself. Arbuscules are cells.
Well, that's a good question. I don't know if the whole arbuscule is a cell. These fungi have very long, drawn-out cells, so I can't tell you if this is one cell or many cells.
Would it be like a chloroplast?
No, it would be like, it is a cell, it's just whether there's more than one cell within the arbuscule. I don't actually know the answer to that. But it is cellular, it isn't a,
Subcellular.
subcellular, yeah, no. The fungus is composed of cells, but they're just very long cells. Does that make sense?
No.
What is confusing?
You see arbuscule inside of it, inside the cell.
So it is a fungus.
Chloroplasts used to be cells, according to, pre-1980,
So chloroplasts are subcellular
and
structures.
mitochondria used to be cells, and those
Yeah.
can just become parasitic and specialized.
Oh, they were,
Organelles.
yeah, and now they're organelles within
Yeah,
plant cells,
and
yeah.
that's where I'm trying to fit arbuscules.
Arbuscules, well, so they're the cells of fungi, and fungi are heterotrophs, so they absorb nutrients from their environment. And so these are absorbing carbon from the host, and they're absorbing nutrients from the environment.
Peggy, I think that part of the deal there is that it looks like there's a single structure there for each plant cell, and I think what's missing is the connection between each of the cells for the overall mycorrhizal.
Oh,
So,
there are hyphae that connect them.
right, so it's all one large organism.
That's right.
And these are subcomponents of it, so it's not the same thing as a bacteria invading another bacteria cell.
Right, okay, thank you. Yeah, this is one organism that's connected. These arbuscules are connected to each other, they're connected to these vesicles, and they're connected to, and they're connected to these hyphae that extend, these thread-like structures that spread out into the soil. So just imagine this is mining the phosphorus and nitrogen, and then it's being transported through these hyphae back into the plant, into these arbuscules that then are exchanging carbon for those nutrients. Is that good?
No.
So these thread-like structures, these hyphae, not only transport nutrients back to the host and water, they also increase soil complexity because many of them have these glue-like substances on their surface that tie soil particles together and form a soil that's more complex, more allows more percolation of water through the soil, and it sequesters carbon. So it sequesters carbon in the soil. So what I'm going to talk about for the rest of our time is work that we did looking at prairies and adjacent old fields, and looking at the diversity and composition of arbuscular and mycorrhizal fungi in the tall grass prairie system, again, in the more eastern prairie peninsula. And then we're going to look at prairie species that, how plants respond to these prairie soil communities that we have. Some of them we have isolated the AM fungi from. And look at how AM fungal inoculum affects plant survival and growth. And then finally talk a little bit about how we can use restoration ecology and agricultural experiments to help our students understand biological concepts. Okay, so we're going to first ask the question, does the soil community recover from disturbance? So when you take a prairie, you plow it under, you grow corn or soybeans on it, or a rotation, and then let it go, what's happening to that soil community? So what we did is we went out and we sampled many different prairie remnants in and around the Chicago area and adjacent old fields or roadsides. We couldn't find an old field adjacent to that prairie remnant or restoration. We looked at the roadsides, we sampled the roadside communities. And what we found was that when you look at AM fungal species diversity, species richness, that they were much higher in the prairies. They were much higher in the prairie restorations and remnants than in the adjacent old fields and roadsides. When we looked at the composition of the AM fungi, so what we're looking at here is one species, Archaeospora trapii, and these are what these spores look like. They're tiny, they're very, very tiny, so it's hard to get a good picture of them. They're clear. And we're looking here at the frequency of occurrence in our sample and the density of the spores within our samples. And what you can see is the density of spores is highest in the roadsides and old fields. So these spores are abundant where there's disturbance. And then in our less disturbed sites, they are much less abundant and density is much less. So we might call these weedy species. These are where we find them with disturbance. And there's other species like Pseudolus dicombrescens where we don't see them with disturbance. So their frequency of occurrence and density is very low in disturbed sites, but much higher in restorations and remnants. So what we learned early on when we began studying these fungi is that AM fungal species diversity declines with disturbance, which we expected, that there are some species we increase with disturbance, and that other species, which we'll call prairie species, are lost with disturbance. But does this matter? Does the plant community care whether or not there's AM fungi that are lost with disturbance? And so we are still doing many experiments exploring different aspects of this. But I'm going to talk to you about work that I began when we first moved to Indiana University. And a lot of this work was done with my husband. We collaborate a lot on our research. And this is the Kankakee Sands restoration, prairie restoration, and it is part of it's a nature conservancy site. And they were tasked to restore close to a 7,000 acre area to prairie and wetlands that had been in agriculture for 10 to 50 years, depending on where you looked at in the sites. So what we did is we used, in our experiment that I'm going to talk about, we used easily establishing prairie plant species. So these plants come in pretty quickly. You just have to sow seed and they will germinate and grow. So we used easily establishing prairie plant species, and we also use difficult to establish prairie plant species. So these species are species that come in, if they come in at all, quite late in a restoration. If you find them in evidence. And so our experimental design looks something like this. We used background soil from the site where we're going to be planting into. We sterilized it in an autoclave. And then we inoculated, our treatments were either inoculation with prairie soil, or inoculation with old fields, so live soil from the site, about 1/10 volume of each, or our sterile background control. So these were our experimental treatments. So we germinated the plants in sterile soil, let them grow, and then we transplanted them into our soil treatments. So this is what they looked like just before we planted them out into the field. This is the sort of the design of the experiment. So we planted into 2 by 2 meter plots. The blue is the plots where we put in inoculated plants that were inoculated with prairie. The yellow were inoculated with old field, and the red was sterile control. Okay, so we had friends help us. They made holes in the soil. We planted. This is what the site looked like right after we finished planting. And this is what it looked like 5 years later. So we had a lot of plants coming in, a lot of prairie plants establishing. Some species, like the Morphokinescens lead plant, only survive in our prairie treatments. So again, we were using whole soil, not A&M. But what we found was that from these studies and others like it, in greenhouse experiments, that whole soil from prairie wetlands enhances the growth and survival of the late succession of the prairie plant species. So we started to recognize that there is sort of a succession happening in prairie. There's these difficult to establish prairie plant species, these late successional species that are benefiting most from inoculation with either whole soil or a fungi. So what I'm going to talk about now is some research that a graduate student of ours, Liz Kogel, did, looking specifically at plant response, prairie plant response to AM fungi that we isolated from the prairie. So she worked with 30 different prairie plant species, and she grew the plants in sterile soil and then with prairie soil inoculated AM fungi specifically that we could isolate. It takes quite a while to isolate these organisms, so this took several years to get these cultures going. And what she found, so what we're looking at here is mycorrhizal response, so that's the difference in growth, the biomass difference between plants that were inoculated and not inoculated. So it's the difference in growth between inoculated plants of the same species and uninoculated plants of the same species. Each one of these dots represents a different prairie plant species. And so we're looking really, these bars are the average mycorrhizal response or growth difference between inoculated and uninoculated plants. And so what we see is that there are plants, what we would consider early successional plants, very early, more weedy species, that on average respond to inoculation with AMF, or vascular mycorrhizal fungi. And then, but the mid and late successional species have the most strong response, and most responsive to inoculation with AMF, or vascular mycorrhizal. So what we know then from this work is that whole soil inoculum from prairie remnant enhances the growth and survival of lakes and sexual species and increases plant community diversity. We know that are AMF responsible? Yes, at least in part they are responsible for both pre-counts and field experiments. So there are other organisms in the community, and Brandon will be talking about some of those in a little bit. But yes, AMF are at least partially responsible for the survivorship and growth in our experiments. Now what I'd like to do is change gears and talk about how we've used restoration how I've used restoration in my classrooms with my undergrads. I have also used it with elementary age children, but I'm going to focus on work with my undergrads because I think it's more relevant to all of you. And this is, I've done this, but Julie and Helen Alexander have a beautiful example of a prairie restoration experiment ongoing here in Kansas. So we identified a site on Indiana University campus that no one cared very much about. That was, this is the mighty Jordan River that runs through campus. And no one cared very much about it, so I asked if I could set up a prairie restoration experiment. And it took a lot more work than I thought. It took a lot of work to get this to happen. And so what we started out was my students in late winter sowed seeds in sterile soil, and then we put them in a refrigerator, a great big refrigerator, so that they would germinate that year. So many prairie plant species need cold, moist stratification to germinate that year, that same year. Once they germinated and grew, then we transplanted them into our soil treatments. Either they were sterile soil from the site or sterile soil from the site that were inoculated with arbuscular mycorrhizal fungi. We killed the turf with, instead of using herbicides, we used black plastic because we were only working in small areas in any given year. Okay. And so this is one of my students pulling up the plastic. And this is planting day. We typically plant during Earth Week. It's an appropriate time. It's just before the semester ends. So we've identified our plots. All the students were involved in all this work. They dug out holes and then we planted in. That students took initial data on the size of the plants. And then in the fall, just at the beginning of the school year, the students measured the plants. And then we analyzed the data to see if there's differences in plant growth between our treatments. And what we're seeing is similar responses in this site as to what we have seen in the prairie restorations in northwestern Indiana. Okay, so now I'm going to change gears again and talk about another program that we started at Indiana University. We got money from a local foundation to work with teachers all over Indiana, for them to work with faculty members to develop experiments that they could take back to their classrooms. And we are obligated, those faculty members who participate are obligated to go into those classrooms and help teachers set these experiments up. And so this is one of our teachers. She was amazing, Carol Fenimore. And this work was done in collaboration with my husband, Jim Bieber, and another teacher. And what we asked, what the question that the teachers and Jim really came up with the question for my husband is, why do farmers rotate corn and beans? And we identified this as a question that was of interest to the teachers and their students because these teachers lived in really rural areas and the students saw corn and beans growing all around them. So this seemed like a good question for them, and it was an interesting question for us because it was looking at the soil community. And so, why do farmers rotate crops? Well, one of the ideas that we learned in undergrad and grad school is that farmers rotate crops because there's nutrient deficiencies involved. So you have corn growing in the same place, or any plant, a monoculture growing in the same place over many years, it takes up nutrients, and those nutrients in the soil are then depleted over time. If you plant another plant in its place, it will continue to deplete the soil, but it might have different nutrients that it requires. So we can imagine that corn growing in corn over time is depleting particular nutrients much more so than if you rotated it with beans. But the other thing is that beans are legumes, so they add nitrogen to the soil. So it could be that rotating the crops allows these plants to recover from nutrient deficiencies that they're experiencing from growing monocultures. Then growing monoculture year after year the same crop. That's one idea. Another idea is its host-specific pathogens. The pathogens accumulate under a particular plant over, if you grow it over years and years and years. Now, this seems to us more likely because farmers use a lot of nutrients. They add a lot of nitrogen, a lot of phosphorus when they're growing plants. And so this is the sort of the question that was set up based on our experiment. This is sort of the experimental design. So let's just go through it. So we had corn and beans that were planted, the students planted, and I'll show you pictures. And they kept some of them at their schools and some of them came back to our greenhouse at Indiana University. And they collected whole soil corn and soil under schools. And what they did is they grew corn and beans either in whole soil, whole corn soil, whole bean soil, sterile corn soil, sterile bean soil, or half bean soil with sterile corn, or half corn soil with sterile bean. Now, in this case, when we look at this, the nutrient levels of both these soils are exactly the same. It's just that half is live, and that live half is corn here and bean here. So we would have different expectations if what's driving this rotation is host-specific pathogens or nutrient deficiency. So what would be our expectations for corn and bean if it's host-specific pathogens in whole corn soil? Which would grow better? Bean, right? What about in whole bean soil? Corn. Okay, in sterile corn and bean. We don't know because it sort of depends on what the nutrient levels of the different soils are, right? And then in the sterile bean, sterile corn, this is live corn with live bean soil with sterile corn, live corn with sterile bean. So what would we expect? Yes, corn would grow better in the live bean would grow better in the live corn soil. But it might be we might not see as strong a difference or a strong growth response, a difference in growth response in the whole soil. But our nutrients are controlled. Now for our nutrient deficiencies, we would expect to see corn and bean growing exactly the same because these soils are exactly the same in these treatments. But they would vary similarly for the plant host-specific pathogens.
How are the soils the same? I'm a little bit confused on the half and half.
Okay, so half the pot is, so we took bean and corn soil. This is sterile, right? And they're mixed 1-to-1.
Right.
So this is 1-to-1, this is 1-to-1. It's just that this is live bean and this is live corn.
But between the two pots, the bean soil, the live bean sterile corn, the bean might have more nutrients than the corn.
So imagine, we just mixed these soils up. So these soils are exactly the same except what's living in them.
From a nutrient standpoint, they're the same. Okay.
Right. It's the nutrient levels that I'm talking about. You got it?
Yes.
Okay,
Thank you.
sorry.
No,
Sorry for not being clear. Okay, so here are the students in our postdoc preparing to plant. Here are some students pretending to look interested in backstage. I asked them to look interested. And so what we see now, here we're looking at the corn, the response to the corn. So this is biomass to the bean soil, which it's not really clear here, is it? But this is bean and this is corn soil. So bean, corn. And so this is the whole soil. So corn grew better than in corn soil, in the whole soil. In sterile, corn grew better in corn soil. So what does that suggest? Well, or lots of nutrients, more nutrients perhaps in the corn than in the bean soil, right? And then here, we see that it's growing better in Y, so this is half, this is the bean soil again, but just half bean, half rye bean, half sterile corn versus the corn. So it is, so what is this suggesting then? Pathogens. Right, and we see a similar trend. It isn't as strong in the bean, but we see a similar trend with the bean. That the bean grows better in whole corn soil. It grows a little bit better in corn soil, suggesting that there's more nutrients in the corn soil. And then it grows a little bit better in the live corn soil. So then the students really enjoy this and the teachers really enjoy working with us. So I'm really happy on both counts that we were able to do this program. But in summary then, we know that AM fungal density and composition changes with disturbance. In prairie systems, we know that whole soil inoculum and AMF and polygrass prairie remnants can enhance survivorship and growth of many late successional plant species. And that students can learn concepts, biological concepts through engagement and research in their local environments. And I'm just going to finish with a picture from our lab. This was at the high point, highest density. Okay, so I'm going to stop there, and I don't know if there's time for questions.
Okay, you're good.
We autoplay. And so I know a lot of high schools don't necessarily have autoplays available to them, but there are places that are relatively close that, you know, like we autoplay the soil for our teachers and brought it to them. Yes.
I'm curious, in the prairie restoration studies where you followed them over a course of so many years, are those still running, and have you seen kind of a spread of the native species now into the plots? Because you weren't physically isolating spread between your plots. Have they?
It turns out that they can spread really quickly.
Yeah, okay.
It turns out that AM fungi can spread pretty quickly. Within probably 3 or 4 years, the uninoculated plots were probably colonized by the inoculated plots. And you can reduce that spread, but we wanted to maximize it because we wanted to increase the rate of restoration. And we definitely saw that compared to other plots, other parts of the restoration where they did the traditional of herbiciding, burning, and then seeding. That we had higher plant diversity in our plots as compared to plots that were started at similar times with that strategy.
So with the goal of taking advantage of the fruits of your research and the research at KU, would it be best then to just trying to think about how I can make this happen at my school, trying to create a small prairie area? So would it be most efficient to just start the plants in a mixed soil with some collected soil from a prairie, a erupted prairie,